So I was down in California for some professional development, and there was a marine station, a marine research station, and they had something called DNA in a tin or DNA in a can. It was a lab in a container about this big, and they could deploy it from a ship into the water, collect water samples, and do DNA testing right then and have the results sent to them electronically. So it's quite remarkable how fast the technology is increasing. So it's all about DNA. So it's about understanding and, as the gentleman said, manipulating genomes, in one case of transgenics, of moving a gene from one genome to another genome of a different species. It's quite remarkable. The human genome was completed in 2003. Oh my goodness, there's been so much that's happened since then. I think that took like 10 years and oh gosh, over $1 billion to do that. Now there are literally hundreds, thousands of genomes that have been sequenced. You can find them online. There's libraries of sequenced genomes online that you can just use in your research. Yeah, so it all depends on advances in technology. Technology like gels and pipetting. That's a technology you're going to learn today at 4:30. So Robin is going to show us how to pipette your DNA in really very small volumes. Yeah, so making recombinant DNA. Recombinant means a new combination. So really, in the large sense, we're all recombinants. You are recombinant because you have some of your mom's DNA, some of your dad's DNA, which has been recombined in you. Recombination happens during meiosis and fertilization. So you can also have recombined chromosomes in that during meiosis, some of the homologous chromosomes cross over, and then you have recombinant chromosomes. So there's different ways of getting recombinants. And in this case, the recombinant is DNA where a gene has been taken from one genome and placed into another. So now there's a recombinant genome. It just means a new combination. It's quite a revolution in biotechnology. So biotechnology technically means using life forms for human uses. That's what biotechnology is. It's been around for a long time. So the Sumerians made beer using yeast. I have not been drinking. Feeling a bit unsteady, but I've not been drinking. So yeah, it's been around for quite a long time. We have manipulated organisms and their genetic components for ages. So one example of that is called selective breeding, where humans have artificially selected certain genomes and selected certain species to breed, and that is a form of biotechnology. The microarray is something that we saw in the personal DNA testing video with Neil deGrasse Tyson, where he was having his personal DNA tested on a microarray that shows a measurement of gene expression of thousands of genes. So this is a microarray like we saw in the film, and the expression, I believe the expression is where the arrays are lit up, fluoresced. So that's a microarray. So there's a couple of different ways we use the term cloning. Cloning can refer to simply to genes. We're cloning or making copies of genes. Gene cloning. we can make multiple identical copies of a gene cloning. And the other kind of cloning is whole organism cloning, which can also be done. But for the time being, we'll talk about gene cloning, and I'll look at whole organism cloning a little bit later on. DNA cloning and its applications. There are many applications, such as gene therapy, for example, even bioremediation. But there's some features that are similar no matter what the application. And we use bacteria, for example, and their plasmids. Now, bacteria are prokaryotes, and you'll see, and you're probably already thinking there may be some problems with that because they're not eukaryotes, but eukaryotes can also be used. So bacteria and their plasmids, I'm not sure if you recall, but well, this is the process, gene cloning with a bacterial plasmid, and some different uses of genes. Let's make that a bit larger. So here's your bacteria. It has its regular DNA, large circular DNA, but also plasmids. Plasmids are circular DNA within the bacteria. And it's very interesting. That's usually the plasmids that are transferred through the sex pili from one bacterium to another, thereby sharing horizontally gene transfer, as opposed to passing genes on to the next generation. That would be like vertical transfer. Bacteria can pass genes on to their same generation and even to different species. That's why Lynn Margulis said that bacteria swim in an enormous gene pool. So here is what is known as a donor. There's a donor, whoops. Donor cell. Here is the DNA in the nucleus, and there's a gene of interest on that DNA that's spliced into the plasmid here. I'll show you how that happens, how that can happen. And then that plasmid is put back into the bacteria with the new gene. Now that's a recombinant plasmid. And then since bacteria divide at an alarming rate, I mean, they're very fast. If bacteria divide every 2 hours, in 2 days, you would get more bacteria than there are atoms in the universe. Not kidding. So, but of course, they're limited in their growth because there's limited food supply. However, you can give bacteria a lot of food supply on petri dishes or elsewhere, and you'll get tons of bacteria, and those bacteria will have the gene of interest. Here it is here. And that gene of interest makes a very particular protein. So those are the proteins. And then you can harvest the protein straight from the bacteria. You can do research on the gene. It could be, for example, a gene for pest resistance. You can insert that into plants, and the plants will have more pest resistance. You can alter bacteria for cleaning up toxic waste. Bacteria like to sequester even toxic things from the environment, and they sequester it, use it for energy in little plastids. You can be producing a protein that dissolves blood clots, and you can produce a human growth hormone to treat stunted growth. That could be a genetic disorder. So lots of uses. for cloning genes. How does it work? Well, you got to know about these guys. They're called restriction enzymes, or sometimes restriction endonucleases. Yeah. Bacteria have restriction enzymes. They cut DNA molecules at a very limited number of very specific DNA sequences, and those are called restriction sites. So it's a restriction enzyme, and it cuts the DNA at a very specific site. It'll make lots of cuts in a DNA molecule. It will yield a set of restriction fragments. And the most useful restriction enzymes will cut DNA in a staggered way. So not straight across the DNA, but between and cut the hydrogen bonds, leaving what are known as sticky ends. They're called that because they will now bind to any other piece of DNA that has a complementary sticky end. They're not particular. It doesn't matter who it is. So like if you had a sticky end from the DNA of the Incredible Hulk, you could complement that with DNA from a duck. So I mean, what I'm trying to say is DNA is not particular with which other pieces of DNA it binds to with hydrogen bonds. I was going to say something else about it, but I can't say that because I'm going to upload this video. So there's another enzyme of interest called DNA ligase. We've seen it before. It binds the Okazaki fragments in DNA replication. It also seals the bonds between restriction fragments. We'll see what that looks like. Here is a segment of DNA with a restriction site. Here. That's your restriction site. Here comes a restriction enzyme, very handy dandy enzyme. It cuts the sugar-phosphate backbones at each arrow, so here and here, and it leaves the pieces of DNA with this end sticking out. That's called the sticky end. And then you can add a DNA fragment from some other source. If it has a complementary sticky end, you're in business. So in some cases, the sticky ends you've cut will just rejoin. That does happen. And you don't get any recombination, but sometimes the bacteria plasmid sticky ends will join at that point. So this is a possible combination. And DNA ligase then seals the strands. And now you've got these pieces of DNA stuck back in there. So yeah, as you were probably guessing, in gene cloning, the original plasmid is called a vector. And it's defined as a DNA molecule that can carry foreign DNA into a cell and replicate there. Does it have to be a bacteria? Yeah, we've taken a eukaryote in this case gene and we put it into a prokaryote plasmid, as incredible as that may seem, but DNA is the universal molecule of life. And it really is. This sort of brings it home just how universal it is. So producing clones. Cloning is used to prepare lots of copies of a gene of interest. It can be used in producing a protein that you want. It could be used in gene therapy. It could just be used in other kinds of research. So here's a human gene inserted into a plasmid from E. coli. E. coli is one of the prokaryotes you find in the large intestine or the colon. And this is where I get a bit lost in this whole thing. but I'll try to make it sort of clear. The plasmid that we're interested from the bacteria contains what's known as an AMPAR gene. That makes the E. coli cells resistant to antibiotic ampicillin. And so as a result, that plasmid can be detected later on when you need to detect it. It also contains this LAC-Z gene, which encodes, this is a beta galactosidase. That's an enzyme that hydrolyzes a molecular mimic of lactose, and it forms a blue product. And that's another way that we're going to identify the plasmids that have taken up the gene of interest. So here it shows isolating the plasmid. This is the plasmid that's been isolated. And the donor, the gene of interest. So there's two segments of this gene, human DNA fragments. And now we have recombinant DNA plasmids. This one has a segment of the gene of interest. This one has another segment of the gene of interest. This one has another segment of the gene of interest. So those are recombinants, but there will also be loads of non-recombinant plasmids. So how do we find the plasmids that have actually taken it up? Because, so what I mean is that here's your bacteria. It's had a chunk taken out of it by the restriction plasmid. Well, it might just, in the end, there's that chunk. The plasmid might just take up its own little chunk there as well. So there's no recombination there. The plasmid just recombined with its own DNA. So it doesn't have a new gene. So we're going to introduce the DNA into bacterial cells that have a mutation in their own LAC-Z gene, so they won't be blue. You plate, that means putting the bacteria on an agar, that's a nutrient dish. It contains ampicillin, which is an antibiotic, and this X-gal, which will be digested by the enzyme if the gene for the enzyme is there. So here's the colony that you've grown. Some of them have non-combinant plasmids with an intact gene, but the colony of interest that carries the recombinant has a disrupted gene. I don't know why. I don't know how that is done, but that is how the plasmid with the gene of interest is identified. So only a cell that took up the plasmid that has that antibiotic will reproduce, because if it doesn't, then it will just die. Colonies with a non-recombinant plasmids will be blue because they can hydrolyze X-gal, but colonies with recombinant plasmids in which the LAC-Z is disrupted will be white. This is just a way of complicated way of identifying which plasmids have the gene of interest, that are carrying the gene of interest. That's what all that is an interest of. So another way is it can be identified with a radioactively labeled nucleic acid probe that has a sequence that's complementary to the gene. That's called nucleic acid hybridization. That's another way of identifying the clone. Let's not read this whole thing. Let's just say, oh yeah, we know how that works. So it's quite interesting, but it's a long process. So I'm not going to go over it all here. You can at your at your leisure. I'm not going to test on this particular method. So what's very interesting is that now that you have these genes, you can store them in a library. So there's a genomic library, and it's made using bacteria with recombinant vector clones. So you can do that from an entire genome. You can cut out the genes from the entire genome, put every one of those genes in a different bacterial plasmid, and store it as a library. And then you have access to every gene that you're interested in. So here's the bacterial clones. This one's carrying one of the genes of interest, this one another one, this one another one. So you can store it here in bacteria. There's also another way, which is using phages. Phages are viruses that infect bacteria. They also have DNA. So you can make a recombinant phage and use the phages in the library. So each one of these has a different gene embedded into its own DNA. So those are called bacteriophages, and they're stored as a collection of phage clones. There's another type of library, which is called a complementary DNA library, complementary DNA. And this is an odd way of making strands of DNA. Instead of using restriction enzymes and all those methods that we just showed, instead, you get the mRNA that's produced by the cell and what's known as a reverse transcriptase that will transcribe the mRNA into DNA. It's called complementary DNA. And then there's another way too. This is the other way. As an alternative to screening the library for a particular sequence, what you can do is you can just get your library to make the proteins and then choose the ones that make the protein that you want. So you can screen those clones for the gene that you want based on detection of its protein, as opposed to trying to find that particular sequence. So because bacteria are bacteria, they're prokaryotes, and eukaryotes are eukaryotes, there are some differences in how they transcribe and translate. Yeah, so there's different promoters, for example, some different control sequences. You can do it, but you have to use a special method of an expression vector that contains a very active prokaryote promoter, or you can use a eukaryote. So nowadays, yeast is very popular. Yeast is a fungi. It's a unicellular fungi, but it's a eukaryote. So what is used often is yeast artificial chromosomes, or YACs. Yeah, so that helps avoid gene expression problems. A eukaryote expressing a gene in another eukaryote is easier, doesn't need so much manipulation. So one thing that we'll be doing in the lab with your DNA, well, you're going to extract your DNA, and then you'll have a little bit of it, a tiny little bit. There's no way you're going to detect that DNA using any method. Although I must say there is a lab at UBC called the Boreal Genomics, I think. If we could, we would go there on a field trip, but we can't right now. However, they have this cool way of detecting little tiny bits of DNA in your blood, even DNA that is kind of indicative of cancer. So being able to detect cancer very early on. We don't have that incredibly expensive technology, so we're going to use another kind of technology in the lab called polymerase chain reaction, or PCR. And that produces loads and loads of copies, many copies, millions of copies. And we're going to, in the lab for you, we're taking out your taster gene for a very specific bitter taste. Um, and when we do that, we're going to use a method of doing that, but then we'll have to amplify that little bit. But we can target that gene. We use primers to target that gene. And we get that from another lab. They have the kit that we can use. And it brackets the desired sequence that we want to get from your DNA. And then we have to use a DNA polymerase, of course, to make loads and loads of copies via replication of your DNA. But we're doing it by heating and cooling your DNA. The heat will separate the strands. This heat-resistant DNA polymerase will come along and make new strands with the nucleotides that we've also put in to the mixture. But man, it needs a lot of heat to separate the DNA. It's too hot for human polymerases. They just denature. So the cool thing is we get this heat-resistant DNA polymerase from I think it's this one is from a bacteria that comes from the mid-Atlantic ridge, the volcanic ridge in the middle of the Atlantic Ocean, where bacteria thrive at that heat. And so they have DNA polymerases that work just fine, just making our DNA too. So this is the procedure of PCR. Denaturation, heat, add that heat, separates the strands. So you don't need the helicase to do that. Then it cools to allow the primer to bond. There's two primers here. We've put the primers into our mixture. Then that heat-resistant DNA polymerase will add nucleotides to the primer, new nucleotides. And then that happens while it's cooling. So while it's cooling, then now you've got your two new strands. And so that was cycle 1. You got 2 strands here. Sorry, cycle 2, you've got 4. Cycle 3, you've got 8. And it doesn't take very long until you've got millions of copies, because we're going to run those on what's known as a gel electrophoresis. And the only way you can detect it is if you have millions of copies. So restriction fragment analysis, what time is it? 4:11, we're good. Restriction fragment analysis detects some DNA differences that affect those restriction sites. And they provide very useful comparative information about DNA sequences. So one way that we can separate these restriction fragments is with gel electrophoresis. So when you use restriction enzymes to cut up your DNA, not all our genes are the same length. Indeed, there are certain fragments that are different lengths for everybody, or I should say certain sections of your DNA that are different lengths for everybody. They're called restriction fragment length polymorphism. So some bits of DNA that you've cut out may be this long, some may be this long, some may be this long. And we can separate them by length. That's very important to remember, because that's what we'll be doing in the lab. So we use gel electrophoresis. So this is a gel here, this square. It has a power source. So here's the cathode end, here's the anode end. And what we have are what are known as wells here. And we put our DNA fragments into the wells. We place our DNA in the well. And maybe we have DNA from 3 different people. Well, the DNA will move in this direction. Why? That's my question to you. Why will the DNA move in that direction? Why will it move toward the positive end? What is it about DNA?
The phosphate?
How? Phosphates, totally. Yeah, phosphates aren't negatively charged. So they will move toward the positive end. But this is an agar. It's kind of a thick gelatinous material. They're not just going to go whizzing. Nuh-uh, they have to go through the gel, kind of snake their way through. So the smaller pieces go faster and farther. So you'll get your small pieces down here and your larger pieces that haven't traveled as far. Larger pieces, smaller pieces. And that's how gel electrophoresis separates your DNA.
Cool. I never got that before. Thanks.
That's cool, isn't it? Yeah, longer molecules be up there, shorter molecules down here. And then what we'll do, so we'll run your DNA on a gel in the lab, and then we'll dye it. There's different kinds of dye. The one that they've used here is, I forget, but it's carcinogenic, so we're not going to use it. We're going to use a blue methylene to dye your DNA, and that works quite well. So yeah, so the reason that we're using it in the lab, and this will all be very useful information for you when you do the write-up. So this DNA lab that you're doing is a write-up later on. So we're doing something called restriction fragment analysis, really. You can compare 2 different DNA molecules, such as 2 alleles for a gene. As we know, our chromosomes are homologous, and for each trait, not each trait, but for some traits, we have 2 different alleles. So here, for example, it's showing normal, I'll make that larger, a normal, I think that's supposed to be an H, hemoglobin allele, here. And here's the one for sickle cell, the mutant allele. So this is interesting because the normal H globin has been restricted by the enzyme here and here. But the sickle cell mutant allele has only been restricted there. So when you run it on the gel, this gel will have 3 pieces to look for. Oh man. And this one will have 2. So then that's what it looks like on the gel. Here you've got the normal allele with 3 pieces and the sickle cell with 2. So that's how you can test whether someone has the sickle cell allele. Yeah, southern blotting. I don't think I'm going to go into that. That's just another way of identifying DNA fragments in a gel. Using probes, you can do that too. You can use radioisotopes, and then they will fluoresce. That's another way. That's called autoradiography. And then the exercise that we're going to do after we watch Robin's demonstration is called restriction fragment length polymorphosis autoradiography. I think that's what it's called anyway. And that's what, I'm not sure if it's exactly the same method now, but probably in forensic labs, that's how sometimes when DNA is extracted from, a crime scene, these methods are used because you're not probably going to get much DNA from a crime scene, sometimes very little, but you can use PCR to amplify it, and then you've got loads of copies, and then you can run it on a gel. And if you've got the DNA from several suspects, that's how you eliminate suspects, for one thing. I can't remember what that's called, but also if you happen to have the DNA on file of the suspect, you might be able to get it there that way. So yeah, they're called RFLPs, fragment length polymorphisms. So on a homologous chromosome, you have fragments that are different lengths. So that's chromosome 1, 2, 3, 4. And for this might have 2 different lengths like that. You might have 2 of the same length there. You might have 2 of the longer length. So those are your probabilities of length. It's very similar to alleles, but these aren't genes. They're just simply fragments that are known. They have names, but they don't code for anything. And they're different lengths. So they're very useful for using with restriction enzymes and gel electrophoresis. And it's called fingerprinting, I believe. Yeah. So southern blotting, I really don't know that much about southern blotting, I'm afraid. But you can map entire genomes using this method. We've mapped tons of genomes of lots of other organisms. Now there's loads of libraries. And I think at the moment that is where I'm going to end this particular lecture. I think that's lots of information.